1929. Berger discovers the EEG, localizes it to the cortex in humans. But and he used it, he was trying to develop a method to study psychiatric disease. I don't think it was very successful, but in 1998 there was a major breakthrough in human physiology, and Nathan Krohn showed that one of the most prominent and salient rhythms in the human brain is ultra-high frequency activity between 60 and 200 or 250 hertz. This signal tracks virtually every cognitive process, and it's been shown to be related to underlying single unit activity. Not as precise as the kind you've seen, more single unit hash from many, many, many cells. Now, the way we get at this is there is about 1% of the population has epilepsy. That's 3.5 million people in the United States. And about 15% of them are intractable, which means they don't respond. And if you can surgically isolate the source, you can really either get control with no meds or get control with meds. So it's a very important technique. And some patients need to have electrodes on the surface of the brain directly, like these different patients with different spacing of electrodes. Some patients, because they have a deep source, maybe in the hippocampal region, need depth electrodes. And more recently, you can see these very fine electrodes. And more recently, techniques have been developed to have little splayed fine wires at the end so you can actually record single units. And I'm not going to, we've just started to do that. I'm not going to tell you anything about that, but it's coming, I think, in humans. Now, you can take these electrodes from many patients and you can link them to atlases. And so you can take a group of, say, 10 or 15 patients and actually lay out in a standard motor, sensory, parietal, frontal regions quite nicely. So you can get control both of the spatial location of the electrodes and you can get control of the temporal parameters because you're recording EEG, which is millisecond resolution. So let's look at a patient with Broca's aphasia who understands everything I'm saying to him, but he can't output speech. Let's check your language out now. Can you tell me what happened when you had the stroke?
Ja, sitting with sorry.
Do you remember, was it in the morning or the afternoon?
TV, TV.
11?
Yeah.
11 in the morning?
Yeah.
11 in the morning. Okay, so he's understanding what I'm asking him, but he has a non-fluent output of speech, sometimes just single utterances, but he's trying to communicate the correct content. So you can imagine how frustrating that is. He has the thought, he can't communicate it. Now, this is Broca's original case, patient Tan, and you can see patient Tan, he's called patient Tan because he just repetitively said tan, 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 and we got the brain, my colleague Nina Dronkers, and we MR'd it. It had been kept in alcohol for 150 years. Looks pretty good for those of you who drink. But if you notice, not only is Broca's area infarcted, but the underlying tissue is infarcted, the arcuate fasciculus and the insula. And if you take a series of 35 of our cases with Broca's aphasia, the brunt of the damage is actually not in Broca's area. It's actually subcortically in the insula and this big connecting fiber, the arcuate fasciculus. So what does that mean? Is Broca's area really engaged when you speak? That's the dominant theory, and I'm going to show you it's not true. If you do an fMRI and I do a really simple task, it's just called verb generation. I say to the patient, I'm going to give you a noun and generate a verb. I say ball, the patient says kick. This happens quite rapidly, and it lays out nicely the classic perisylvian language areas for comprehension, Wernicke's area, Broca's area, motor when you speak, but you don't know the temporal sequencing because the MRI is a sluggish response. If you use electrocorticography and focus on high-frequency activity, and this is work by Eric Edwards, what you're going to see is a beautiful evolution of language. This is 2 seconds of language stretched out over 10 seconds, or it goes by too fast. And you'll just see it unfold, perceive, ball, select, verb, produce the motor program, we're not even at a second, drive your articulators, and then since you said something, you redrove your auditory cortex. So this technology can precisely map cognitive function in action. Now, Dean Flinker used this to really assess whether Broca's area is active when you speak. And he did a variety of tasks, reading and speaking, And it's pretty clear. If you look at this high-frequency brain activity, there's robust activity in auditory area. There's robust activity in Broca's area, which is actually linked temporally to auditory areas, but when you start to speak, Broca's area actually shuts off. So the dominant theory, and nearby motor areas are active, so the dominant theory about Broca's area being involved in the production of speech, it's not true. Now, the beauty of this technique, and this will become important when I show you our attempts to develop a speech prosthesis, it's reliable at the single trial level, sort of like, raster scans in animals. So each one of these is an individual sweep of one of 7 patients doing a speaking task. You can see robust activity linked to the auditory areas, Wernicke's area. You can see Broca's area linked to this also with a delay of 150 milliseconds, but here's when you speak. It's pretty clear when you start to speak, Broca's area shuts off, but nearby motor areas are active. So that shows you something about the power of the technology in addressing old questions in language. How about the hippocampus? We've seen elegant work on memory and spatial ability and linked to the hippocampus. Most people don't think the hippocampus is involved in language other than to recall or store words or phrases, etc. Turns out that's wrong too. If you do a very simple experiment, he locked the door with blank, then you name key, and you have another sentence is she walked in here with key. This has semantics, this has context. It narrows your search for this word, and every patient gets faster. This is faster reaction time when you have a semantically, contextually appropriate lead into the key as opposed to not. And we know now that if you have an infarction or damage in your hippocampus, your language seems normal, except if you really examine the high-level semantic extraction, the patients are off. So what happens if we look at the hippocampus with an electrode in the hippocampus as patients are doing this task? Well, what you find is actually just like in many other studies of the hippocampus, as the semantic context of the sentence builds up, so it's narrowing the choice of words, you get rhythmic hippocampal theta virtually in every subject. So again, this approach, this human neurophysiological approach allows you to kind of address, challenge, or better define classic questions on how the brain is actually organized to do behavior. Now, one minor thing I want to mention, and this is one of our main areas of research, and I won't have time to go into it, is these networks for language are rapidly established, and they have to become coherent. And the way they become coherent, and for those of you not familiar with coherence, here's coherence at a playground. First kid gets pushed, kid swinging at a certain frequency, mom or dad pushes the second kid, they're out of sync. But the third kid's pushed, but these kids want to get in sync because they want to look at each other and laugh as they're swinging. Well, this happens over many, many seconds on the playground. And there they are in sync, but it happens in your brain in 100 milliseconds. So these networks evolve very rapidly change, and the idea is if you put 2 brain areas in the same coherent state or the same excitability threshold, it's much easier to transmit the coin of the realm, which is single unit action volleys between brain areas. Now, I'll just show you a very quick, one small, and by the way, it's been shown for language, memory, attention, executive control, these coherence networks. I'll just show you one brief example. This is, again, you've seen pictures. Here's our amygdala in red, our beautiful hippocampus, much research linking the amygdala to emotional processing. So how do these 2 areas interact as you're processing emotional versus non-emotional stimuli? Does the hippocampus drive the amygdala, or does the amygdala drive the hippocampus? So here's the stimuli, very simple. There's neutral landscapes that the patient sees. And there's fearful faces that the patient sees. So, the emotionally laden, not emotionally laden. So the question of what happens, we have electrodes in the hippocampus. Yellow is the amygdala, blue is the hippocampus, here's the head, and here's the tail. Each one is an individual subject. And actually what you see when you do this very simple experiment and you use these coherence metrics, what you find, in fact, is that only in the fearful condition does the amygdala actually drive the hippocampus. And this is actually why it's been known for many years that an emotional event is better remembered than a non-emotional event. It's just, classic. So this is just the infrastructure to let you know these signals are very powerful and they give you insights into human behavior. So let's talk about the speech prosthesis. How could we get there? So this is, again, some work by Dean Flinker, who did the Broca's work, and he showed a few years ago that, in fact, here's a grid. And by the way, some of this data is obtained in the operating room, which is a pretty tough environment. The patient has to be awake if you're going to map language, or you can't map language. You can see the brain pulsing, the cardioballistic effects, etc.
You feel some vibration, Susan.
Okay, so what he showed with these high-density grids in auditory areas is that you get this rich mosaic of activity, and here is 9 electrodes right there. You can see one electrode, this electrode responds to hearing, this electrode only responds to speaking. So the first, what you know right away, this idea that there's a blob of language area is just ridiculous, right? It's a very complex interleaved mosaic of nearby cell assemblies that are doing different things. And it's reliable, actually, at the single trial level. Here's a high gamma burst to hearing. Here's a nearby electrode with a high gamma burst to speaking. Now, then if you look at this grid and you take a phoneme, let's say ba, and you take a word, let's say tree, of the same length of time, what you see is this very robust grid of activations for phonemes, but a much more sustained activation pattern for words. So we had this as an infrastructure. We knew there was a rich tiling of activity, and we said, can we extract information from that might lead us to be able to build a prosthetic device? So again, this is a patient, and we're just playing phonemes to the patients, and we're using these high-density grids. They're 3 millimeter spacing, they're 256 channels, and you can see this very robust activation to phonemes and auditory structures. And by the way, it's in both hemispheres, left and right hemisphere. So first, we used it to address a phenomenon called categorical perception. So the question is all the world taken in as one continuous stream, and then later on you put it into buckets, dog, cat? Or do you actually take in the world as categories? Now, categories, doing it the second way would speed up processing and require less neural resources. So this has been known since 1956. You're going to hear ba, das, and gas. They're absolutely the difference. There's 14 gradients of sound that are in exact equal steps, but that's not what you're going to hear. Your brain is going to separate it into 3 categories. So just listen.
La, 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 la.
Did people perceive 3 categories? That's called categorical perception. And Rieger and Eddie Chang, who now has a fantastic lab of his own at UCSF, showed quite nicely that your brain is doing this bucketing, this categorical perception, within about 110 milliseconds in auditory regions. And they're clustered, right? There's clusters of bas, das, and gas electrodes, interleaved. Different colors are related to different ones of these phonemes. So again, this interleaved mosaic of activity. And so we had this, we knew that we had phoneme representation. We knew we could decode them. We could actually tell what phoneme the person heard. Was it a ba, or was it a da, or was it a ga? So then we did some work on how plastic is this system? How rapidly does it change? And I'm going to play another sound file for you. And I'm only going to play one of the conditions. You're going to hear a phrase that has had the linguistic features of it taken out by a technique that's developed from birdsong by a colleague, Frederick Tunison. You will not be able to understand it. Then I'm going to play the phrase, the right phrase, and then I'm going to play the exact same scrambled phrase again. And what you're going to hear, I think, is that all of a sudden, the scrambled phrase makes sense. And of course, you have appropriate controls with pink noise, so it's not just basically repetition, etc. And what you find, if in here you have no context, which is noise, you have no ability to detect any of the words. If you have a little context, but it's the wrong context, the wrong phrase, you get a little better. But if I put the right context in, the phrase that I've taken apart, you're going to have a tremendous improvement in discrimination of words. So listen to the sound file.
Bagpipes and bongos are musical instruments.
Bagpipes and bongos are musical instruments.
Bagpipes and bongos are musical instruments.
That's the same sound file.
The high security prison was surrounded by barbed wire.
The high security prison was surrounded by barbed wire.
The high security prison was surrounded by barbed wire.
The full moon shone brightly that night.
It's crazy, right? I mean, and it's happening in a second.
Move the garbage nearer to the large window.
Something I forget to do. But anyway, and this is work done by Chris Holgraff, really beautiful work. So what's going on, right? That's the behavioral effect. It's been known for a long time. What Chris showed is actually the spatial temporal receptive fields of ensembles of cells, probably 100,000 cells, maybe more. They change from being not speech to speech after one presentation of speech. And the spatial temporal receptive field just simply means here's frequency, here's time, and here's where a particular ensemble, and it's also been shown with cells in ferret, individual cells, it fires. And what happens in this particular phenomenon in the human cortex is this is tuned, these are cells after you've heard it the second time, the scrambled speech that you perceive, the spatial temporal receptive fields switch to be more speech-like, and it's very, very rapid. So these kind of infrastructure things made us encouraged that we could maybe get out of speech prosthetics. So what are we trying to do? You're trying to read the notes of the cortex. So if you're Beethoven and you're deaf, and you watch somebody playing the piano, you can reconstruct in your head, I'm sure he could, precisely the musical melody that the person was playing. So that's what we're trying to do. We're trying to assign to each electrode at a specific time a certain frequency. Now we've extended it to phonemes and we'd like to go up the linguistic chain. I'm going to show you our initial work with just frequency. So just like this piano key at some point in the phrase might fire and be a C, we're trying to ascribe that to auditory electrodes. So this was work done by Brian Paisley. We presented patients with 100 different words. We had electrodes over the proper area. We recorded their intracranial EEG directly from their auditory areas. We came up with reconstruction models, and we then tried to reconstruct, let's say this is football. Now, you know, when you have models, they are pretty unconstrained. You're not sure your model really means anything. So what you have to do is hold out data from the model. So you don't use all the words to train the model. Now you represent the words and see whether the model distinguishes between two words. So let's say the word is banana versus, and you have it versus orangutan. 50-50 would be, you know, you're not anything. You're not succeeding at all. It turned out Brian found a 91% accuracy that we were able to decode what words you perceived. And I said to Brian, make a sound file. And he said, no, that's not scientific. And I said, do you want to get a job? So he said, okay. So he made he made a sound file. It's a true story. I don't know. No one seems to listen. I don't know if people have that problem in their lab. It's very difficult. It's good. So here, actually, you're going to hear 4 words that the patient heard: Waldo, structure, doubt, property. And then you're going to hear the word reconstructed directly from brain electrical activity, played back on a speaker. And you can decide whether there's any strength in this approach. And the methods I'm not going into, they're obviously, machine learning and multiple regressions, etc. Okay, so just listen.
Waldo? structure, doubt, property.
So I think you could hear that we could actually reconstruct what you heard from your brain and play it back. And this is the first pass with 4 millimeter spaced electrodes. And we know there's independent activity at about 1.2 millimeters. And we keep pushing the boundary and trying to get tighter electrodes to improve the reconstruction ability. So this is nice, pretty nice sound file. But the real question is, can we now do what we wanted to do? Is to make a speech prosthesis. Could we implant an electrode, a wireless electrode, over the auditory speech areas of someone who can't speak but knows what they want to say? Oh, by the way, you can do not just words. Here's single phrases reconstructed from the brain.
That's my fire escape.
Sometimes not so good. Not bad, but not so good.
From some of your friends at the camp.
First one pretty good, second one not so good. This is older data, but I wanted to show you, nothing's ever as perfect as it looks in the paper. So in terms of the imagination, we approached it from 2 levels. The problem with imagination is knowing when somebody imagines. It's really hard. It's as hard as humans in a way as it is in mice. You don't really know when the onset of the imagination is, so we try to have some external cue to drive it. And one of the things we did, since work is done in the United States, we actually have a speech from one of our, let's say, better presidents, Abraham Lincoln. And this is the famous Gettysburg Address, 4 and you'll hear, and there's 2 conditions. The ticker tape is going by, so we thought the ticker tape would give us a time code to look for the imagined speech. In one condition, the patient says the ticker tape. In the other condition, they imagine the ticker tape. And we have the data from both, and we can see, can we reconstruct her or his imagined speech?
87 years ago, our fathers brought forth on this.
Okay, so you get the idea. The other thing we've done, which is even simpler, is to simply do experiments where you have an auditory cue and they listen to, let's say, baseball. Then they get a visual cue and they have to imagine baseball. And then they get another visual cue and they'll say baseball. So we can decode them hearing it. We can decode when they say it and drive the auditory cortex. And we can use these models to help us extract the imagined activity, because that's the goal. Does it work? The answer is yes. This is work done by Stephanie Martin, who's a bioengineering student. She just got her PhD in EPFL. And what you can see, I've already shown you, we can accurately decode what the person heard. We can also accurately decode what they imagine. Significant in all the subjects. The areas of the significant decoding, when you look at the brain and where the electrodes are, not so surprisingly, are the classic players in the superior temporal lobe. Even some in this right hemisphere case. So this is very exciting. It's not at the level we'd like it to be. We have probably several years of research to get it, so not only can we statistically say the person is imagining baseball versus football or dog versus tree, it's not at the level where I could play it to you right now like I could play the reconstruction of the actual driven activity in the auditory cortex, but we're making progress. So what about music? If we could do language, we should be able to do music. It turns out the patients are in the hospital for a long time, 3, 4, 5, 6, 7 days. You're being monitored, clinical stuff, research stuff if they agree, and they get to pick songs, whenever they want to hear music. And it turns out a lot of them pick Pink Floyd. So we had 28 patients who listened to Another Brick in the Wall.
Just a brick in the wall.
And everybody knows this song.
All in all it was, all just bricks in the wall.
Okay, so first, who's a Pink Floyd fan here? Anybody? What is wrong with this slide? Tell me. What's wrong? Anybody? It's the wrong album cover. This is from The Other Side of the Moon, but I thought the spectral decomposition fit better with the with the talk. So anyway, So the nice thing about Another Brick in the Wall, it's 4 minutes long. So that means we could take the first 2 minutes of the song and come up with a model for decoding music, and then apply that model to the last 2 minutes. So here's the results. Again, we had 28 subjects, lots of electrodes. Nicely, there's so many electrodes that overlap. The inner electrode spacing is now 1.1 millimeters. Out of all those electrodes, what I'm going to show you, the electrodes that are informative for the decoding of music are actually, not so surprisingly, most of them in auditory regions, although there are ventral premotor areas also that contribute, and that's a whole nother story about the role of the ventral premotor system in language. So here's the results. So I'm going to play 2 sound files. One sound file is the actual last 2 minutes, so I won't play all of it, of Pink Floyd. It sounds a little grainy because we were only able to go up to 7 kilohertz in our reconstruction because it just becomes combinatorially intense. That problem will fall with improving computer power. So first, here's the actual song. Music, whistle, language. Okay, so can we reconstruct it? The answer is yes, not so surprisingly, because if we could do language, I think music is even in some ways an easier frequency space. So that's Pink Floyd being played directly from electrical recordings in the brain. And I think, why even do music? Well, we had the data, so why not look? But music also has prosodic elements. And Eddie Chang, who I mentioned earlier in the talk, just published a paper in Science last week showing spatial temporal receptive fields to prosody in human cortex. So if you really want to have a speech prosthesis, you'd like it to have not only, I'm hungry, it might be, I'm hungry, or I love you, I love you. You'd like to have a prosodic element. So we think it's an important part of the program. So finally, and I'll end with this, what about music imagination? So we had a young man, 15-year-old man, who contacted us. His father's actually a biology professor, and there's no human subjects issue here because he actually contributed so much to the study, he's actually on the paper. And he said, I'm going to have epilepsy surgery, and I know you're doing music research. Can I participate? And we said yes. And he basically practiced for 6 weeks. He's a very accomplished piano player. And by the way, for those of you who think epileptic patients are weird and can't do things, that's absolutely not true. That's just a myth. He basically practiced a Brahms piece and a Beethoven piece for 6 weeks on his keyboard. And he basically practiced it hearing the sound, and then he practiced it hitting the keys and imagining the sound. So this gave us a very beautiful time code for when he was imagining, just like the ticker tape or the controlled hear, listen, etc. that I showed you. And the results were very, very nice. This particular man, a young man, basically, we could get electrodes in his auditory region that responded both to hearing the music as he played it and to imagining the music as he played it, so that you can see these 2 electrodes are directly or very nicely overlapping. Some electrodes only responded to the perception of the music, but overall, when you look at his perception areas, again, extensive temporal areas, not surprising, ventral premotor areas, and this is just a blow up of this little block here of his neural activity in this high frequency band, which is the one by the way, I should reemphasize, all this reconstruction is using the high frequency band of your activity, which is, we use 70 to 200 hertz. That's everything I've shown you has been using that particular subcomponent of the human electrical activity in the brain. You can see beautiful activity here, And you can see when he imagines very similar areas being activated, and when you blow it up, you know, not as robust. The imagine is never clearly as robust. Part of it may be related to timing, we don't know, but it's quite clear that we can reconstruct imagine speech and imagine music. And that's kind of setting an infrastructure that if we can continue to develop this and get better at the computational approaches and the technical approaches, a little bit like Edvard was saying, more electrodes, right? Higher density electrodes, more information, more extension just from frequency to phoneme, to syntax, lexicon, etc. I think the door is open now that maybe in the not so distant future, if you had a loved one who had a Broca's aphasia and couldn't speak, they may be a candidate for an implantable wireless device, and they could actually communicate with you. And that's the ultimate goal of this particular line of research. And of course, the students and the postdocs and the grad students involved in this project, the ones I starred actually contributed, have absolutely been tremendous. So I'll stop there and be glad to take any talks, any questions.
Thank you. Thank you very much, Professor Knight. We're now going to try something different. Excuse me, if I can just walk past you, because you won't be needing this. Let me see. Catch it. OK. Hi. Now hold it up so you have the black part, not that part, but yes, exactly. Hold it under your chin, like if you were trying to shine a light up on your face, and tell me, what's your name?
Axel.
Excellent, this works. Axel is now trying a catch box, which is a flying microphone that we can pass around the room so that people can ask their questions right into it.
Like a beach ball at a party.
Yes, like a beach ball, but with a microphone, which is super. Axel, do you have a question for Professor Knight?
No, I'm sorry.
That is all right. We're still very happy to have you here. Any other hands in the air? Any other people that have a question? Over there, which means that Axel will try to throw it there, and if it doesn't go all the way, somebody else will help. Super, thank you.
All right, hi. So, my name's Andre, and I'm wondering, how will this prosthesis work? Will it work via generating the speech or helping the patient figure out how to plan the motorics of
the target is to be used when they're generating internally generated thought and language. That's the definite goal. Now, you could flip the process. One other line of work that we're beginning is if you know the topographical organization of language in these ensembles, theoretically you can stimulate, and you could have a central hearing aid for people who can't use a hearing aid anymore. So there's another potential line of research. I will say there was a funny thing. We have a kids journal where people write articles for kids, and we have these live reviews where the kids are, the scientist, Stephanie, by the way, was presenting to 6 kids about her. Where is she? She's up here somewhere. Where is she?
Up at the top?
Where's
Stephanie?
Stephanie
At the top?
There.
Oh,
okay.
there, that's Stephanie.
So she's very proper, Swiss, good. And she was talking about this Imagine Speech project. And this 10-year-old kid says to her, and he's really cute, Wyatt, he says, so if you got this speech thing and it's decoding what you're thinking, suppose I have one implanted in me, and I'm looking at you and I say, I'm thinking, boy, your hair looks weird. She's going like, my hair. The trick is turn it off, I guess. But yeah, we want to, it's pretty simple. We want to be able to when you have a patient with a neurological problem, I don't care what it is, if you give them any help, it's a big deal. So if you have someone who's hemiparetic and they can't open their hand, try to open a soda can with one hand. If you can simply get a device, and this is a lot of the work in brain-machine interfaces and motor, we just decided to do language. If you can just get them to open and close their hand, doesn't sound like it's much, but it's a huge thing. So that's what we want to do. Give something to the patients that'll make their life better.
Thanks.
Yes, we're really hoping for this to come true. More questions? Was that a hand in the air? No? Oh, over there. Over here. So we need the catch box to move all the way through the room. There you go. And a little bit more. The gentleman, person who wanted it, hold your hands up. Perfect. Thank you. And then you, yes, that's the right way.
There's a microphone here, yeah? Good thing. This could also be used for hearing impaired people. Have you been looking at that?
Well, that's what I mentioned. If we had someone who had severe hearing loss, and they're not really a candidate for cochlear implant, the idea would be we'd have a central hearing aid where whatever sound is being played to them would be broken down and sent into channels in these same electrodes that we're decoding speech from, and we'd stimulate in a spatial temporal pattern that we might be able to produce a hearing aid. I have a postdoc working on that. I mean, it's a real long shot, but you never know with technology and computational and better electrodes. So yes, the answer is that's on the plate. It's not at the top of the list.
Thank you. More questions?
There.
We can have one last one. Yeah, over here. So if you throw it this way, yes, give it a go. That's right. And it's, it can take that, so just get it up again and throw it. And one more last bout of throwing. There you go.
Nice.
And now it's upside down, so turn it again. Perfect.
Okay, so there is a sort of coma, a state where you can think you're awake, but you can't move your body.
Yeah, and in
can
fact,
this be used for that too?
Well, it could, certainly. I mean, Colin last night brought that up, and people who are locked in, who've had usually a pontine brain infarct, and they can't speak, they can't move anything, they can only move their eyes up and down. That'd be a definite targeted patient group. It might even be a targeted patient group for people in the minimally conscious state who are not. Coma goes from deep, usually die, to vegetative state with no contact, and your eyes are drifting around, to minimally conscious. So minimally conscious could be another target population that you could help. There's even, this is pushing it a little bit, but there's unfortunate kids with severe neurodevelopmental disorders that have very bad speech apraxia, and they can't speak. So I don't think we'd really be looking at kids first. We'd probably start with we're talking with a group in Germany of starting with a cohort of 4 ALS patients and seeing if we can get something to work with them. But yeah, I think you're right. If you think of the spectrum of people, it's pretty big that this could be applicable to. Thank you for that question.
Thank you very much. Thanks for all your questions, and thanks to Professor Bob Knight.